article, can I go?
Mm-hmm.
I did the article about pollutants increasing sonic complexity and the volume of the brain area HVC, not P, in the songbird. So this study talks about the behavioral and physiological changes seen in male starlings exposed to EVCs, and the behavioral changes are changes in their song length and complexity of song, where the physiological changes are lower immune function and HVC enlargement. And then there were 3 different groups tested. One was a control group, one was a group that was just given estradiol, and then there was a mixed group, which was given a mixture of different EDCs, which they determined from actual testing they did on affluent filter beds, which I'll talk about later. And so these male songbirds were affected by environmental pollutants. They were exposed to estrogen mimics through invertebrates which they ate, which eat organic sediment, which is used as filter beds to clean affluent runoffs. So this is how they purify the water, but they ended up exposing all these invertebrates to the EDCs because of the organic sediment started micro and macro environments. And the HVC is the high vocal center, and it's a nucleus in the brain responsible for learning and the production of songs in male songbirds. So a male song is a sexually selected trait by females, and this is by how well a male sings, and it acts as an indicator of male quality. Song production is controlled by neural pathways, which are regulated by the endocrine system, and estrogen plays a role in masculinization of the song center, which I'll get to later in this presentation. HVC is the only place in the song pathway with estrogen receptors, so it's going to be most susceptible to the estrogenics, which are found in the affluent runoff. And exposure, like I said before, exposure is coming from the affluent runoff because of all the invertebrates that are living in the organic soil. And this study tested immune function. They tested cell-mediated response and humoral response. And for their song production, they looked at length, number of bouts, the repertoire of a bird, and the complexity of their song. And then for the neural development, they looked at HVC size in the brain. So for methods and materials, they looked at what the starlings ate in their winter grounds, which is where they would just nest for the winter and then they'd go off to the breeding grounds. So they looked at 20 different sites, and what they found was that earthworms were the most abundant prey eaten. So they tested the earthworms for what EVCs might be contaminating them, and therefore contaminating the birds. And they used 5 different places to get the mean exposure levels. And as I said before, there are 3 different groups. And then the birds were dosed from October 2004 to April 2005. And what they did were they were given 5 doses per week of mealworms, which were injected with either peanut oil for the control, estradiol for the E2 group, and then a mixture of 3 or 4 different estrogenics, including estradiol. And then they were allowed water ad lim, and they were also allowed insect pate, which is quite exquisite, I've heard.
We may all be eating it.
Yeah. So then in 2005, they tested blood samples for testosterone levels and hematocrit levels, which is a proportion of blood volume that is occupied by red blood cells. And then the cell-mediated immune response was tested by injecting the bird's wings with PHA, and then they were measured before injection and after injection to see how much the swelling went down in the wing. And they determined the cell-mediated immune response from how well the swelling went down, and it said after 24 hours, all swelling had subsided in all the birds. And I wasn't quite sure how they did the humoral response. They took blood and tested the plasma, and what they did was they injected sheep antibodies into the bird, but I wasn't really sure how...
Sheep red blood cells.
Oh, sheep red blood cells. And I know humoral response is antibody response, so I would assume that they were testing to see how many antibodies were in their blood after they were exposed to these sheep's red blood cells, and that's how they determined it. So they recorded song data, and they analyzed the length of the time spent singing, the number of song bouts, the duration of each song bout, and the repertoire size of the bird. And they did this by placing one male and one female in 2 different cages, and just they let the bird sing for, I think it was, they recorded twice for 3 hours, and then I want to say they did it for 5 minutes, and then to actually test which ones the females preferred, they repeated it for 30 minutes, I think it was, and then it ended up being like a 3-hour sequence of a bird song. And then, so like I said before, testosterone levels were tested, and the brain area was measured, as well as the size of the HVC in the male birds. And then song preferences of females were observed between the control and the estradiol group, the control and the mixed group, and the estradiol group and the mixed group. And how they did this was they put female birds in a long aviary and put 2 speakers hidden on the left side and the right side, and then played 2 songs from the 2 different groups and saw which ones the females went to more. This graph is about the immune function of the birds, and as you can see, the clear one is the control, the middle one is the E2 group, and the one on the right is the mixed group. And as you can see, there's a really significant difference between the control and the mixed group, and this is for the humoral response, and then this is the wing swelling, so this is the humoral, and then this is the cell-mediated response. And in the top graph, you can see that the E2 group actually had the least effect on the swelling, but in the humoral response, the mixed group was the lowest, but they're both significantly different than what the control group showed. This is the song production of males, and I'm really sorry you can't read this. I had a really hard time bringing the graphs into my PowerPoint. But as you can see, in all groups, the mixed group is the largest and most predominant one. So that means it had the longest song length, it was the most complex songs, their repertoire was higher, and they had the most bouts of songs. This one is the HVC size, which is the nucleus in the brain. And again, you can see, compared to the control, the mixed group is the highest, but you see that the E2 group does have a difference from the control, but it's not significantly different. And then I wasn't quite sure what they were showing. I know this is an actual picture of the HVC nucleus, but I wasn't sure if they were like looking at the dot